Hello and welcome to some online class. Friends, today we shall discuss about LAC operon. The operon model, a negative control mechanism, was developed in 1961 by Francois Jacob and Jacques Monod to explain the regulation of genes that are involved in the utilization of lactose in E. coli. In prokaryotes, genes with related functions often are present in a coordinately regulated genetic unit known as operon. Operons can be divided into two different groups: inducible operon and repressible operon. In an inducible operon, the free repressor can bind with the operator for turning off its transcription, whereas in a repressible operon, the free repressor cannot bind with the operator. So first of all, the free repressor bind with a co-repressor. After this, the repressor-co-repressor complex bind with the operator for turning off the transcription. Now we shall discuss about the various components of the LAC operon. The LAC operon contain three structural genes. The three structural genes that are present in the LAC operon are Z gene, Y gene, A gene. And the three operators that are also the part of the LAC operon are O1, O2 and O3. O1, O2 and O3, they are the three operators that are present in the LAC operon. Another specific region is promoter region. So three operator regions, one promoter region, and these three structural genes together they constitute the LAC operon. In case of LAC operon, a regulatory gene designated as I gene is also contiguous with the LAC operon. This I gene has its own promoter region that is known as PI. So PI is the promoter of this gene. Next, out of three structural genes, the Z gene encodes the enzyme beta-galactosidase. Beta-galactosidase break down lactose into glucose and galactose, whereas the Y gene it encodes beta-galactoside permease. This beta-galactoside permease is responsible for pumping lactose into the cell. And another gene is A gene. A gene encodes an enzyme known as beta-galactoside transacetylase, whose function is not clearly known. So these are the various components of the LAC operon. In the operon model that was proposed by Jacob and Monod, it had only one operator, which is now designated as O1. Subsequently, two additional operator regions were also discovered. These two operator regions, which were subsequently discovered, are designated as O2 and O3. So the LAC operon contains three operators: O1, O2 and O3. The LAC operon is a negatively controlled inducible operon because these three structural genes are expressed only in the presence of lactose. The LAC operon is under the control of a regulatory gene known as I gene. The I gene is a regulatory gene that encodes a protein known as repressor protein. This repressor protein is 360 amino acid long. The active form of the repressor that can bind with the operator region is a tetramer. This tetramer contains 4 copies of the product of the I gene. So this tetrameric form of the repressor bind with the operator region and prevent the enzyme RNA polymerase from catalyzing the transcription of these structural genes. In uninduced state, a few copies of the three different enzymes are also synthesized. This gives a low level of background enzyme activity. This low level of background enzyme activity is necessary for induction because for the LAC operon, inducer is the allolactose. Allolactose is derived from lactose in the presence of enzyme beta-galactosidase. So after its formation, the allolactose binds with the tetrameric repressor and release the repressor from the operator region, after which the structural genes can be transcribed. This is about the induction of LAC operon. So LAC operon is an example of negatively controlled inducible operon. Now we shall see as to how lactose induce the LAC operon. As you already know that there is a gene known as regulatory gene, and that regulatory gene encodes a protein known as repressor protein. The active form of the repressor is a tetramer. In the absence of lactose, this tetrameric form of the repressor bind with the operator region, thereby preventing the enzyme RNA polymerase from catalyzing the transcription of these structural genes. But in the presence of lactose, the lactose is converted into allolactose in the presence of enzyme beta-galactosidase. In the presence of enzyme beta-galactosidase, lactose is converted into allolactose. The allolactose is the actual inducer of this LAC operon. The allolactose bind with the repressor and convert the repressor into an inactive form that cannot bind with the operator region. So, the transcription of these three structural genes proceeds as usual. This is the process as to how lactose acts as the inducer of the LAC operon. Now we shall discuss about another phenomenon known as catabolite repression. The catabolite repression also play an important role in the expression of the LAC operon. It has long been known that the presence of glucose in the nutrient medium prevents the utilization of other energy sources. So when glucose is present in the nutrient medium, in that case only glucose is utilized. So glucose is preferentially utilized as the source of energy. This phenomenon is known as catabolite repression or glucose effect. The catabolite repression of LAC operon and other related operon is mediated by a protein known as catabolite activator protein. The protein known as catabolite activator protein along with a small effector molecule known as cyclic AMP. Cyclic AMP is cyclic adenosine 3,5 monophosphate. In the LAC operon, the promoter contains two different binding sites. One binding site is for binding with CAP-CMP complex. Other binding site is the site where the enzyme RNA polymerase bind. So one binding site is CAP-CMP binding site, another binding site is RNA polymerase binding site. So when the CAP-CMP complex is present at its binding site, then only the LAC operon is normally induced. But in the absence of CAP-CMP complex at its binding site, this LAC operon cannot be induced because in the absence of CAP-CMP complex, RNA polymerase cannot bind with this promoter region. So in the absence of RNA polymerase, the structural genes cannot be transcribed. So how this is possible? There is an enzyme known as adenylate cyclase. Adenylate cyclase is an enzyme that catalyzes the formation of cyclic AMP from ATP. Glucose, it inhibits the activation of the enzyme adenylate cyclase. So when the enzyme adenylate cyclase is inactivated, sufficient amount of cyclic AMP is not formed. In the absence of cyclic AMP, the catabolite activator protein alone cannot bind with the CAP-CMP binding site. So when the CAP-CMP complex is absent at its binding site, in that case, the RNA polymerase also cannot bind with its binding site. So in the absence of binding of the enzyme RNA polymerase, the genes cannot be transcribed. So the LAC operon will be repressed. So friends, this is the mechanism of catabolite repression. For more updates, please subscribe my YouTube channel Sam Online Class. Thank you very much. See you friends again. Bye bye.